Thanks a lot. We'll try to make 2 talks in 20 minutes. And so first talk will be on behalf of Viorel Munteanu from Romania University as well as Moldova University, who was not able to come. And I will talk about reusability of Omics public data. And so first thing about Sage. So it was great to see presence of Sage. So we try to do open data, and we try to think about reusability and radical collaboration. And as you saw, as well as synthetic data, so many interesting things. So I would like to start and kind of set up the stage and say that I think many of this audience agree that we are moving to the data-driven world, where many things which were not possible before are possible now. And I think people who work with the data, like the people like we are in the audience, I think we're shaping the future, and we are shaping the way we're going to see biomedical research. And I think data science and particular data sharing is crucial concept. So if you think about data sharing and in general data generation, we went far away to generate quite a bit of data. So this is, I think, already outdated slide, but we have millions and millions of samples sitting in the public domains, which many of them are publicly available, meaning that you can go on the website and download the data, and perhaps there is decent metadata and do that. So why that's important? So beyond the purpose of transparency, that the data is there and you can go and check the results which were based on that data in the regional publication, can go and do something else. You can do what we call secondary analysis of the data. So you can do the attempt to reuse the data again, and with the hope to find some novel biological discoveries or some interesting insights. And there are a few reasons to do that. You can do some sort of meta-analysis, combining different studies. You can also try to do better bioinformatics, or you can repurpose the sequencing technology, meaning you will analyze it from a different angle or perspective. So why it's important? Why it's important to reuse the data? So it's increased scientific impact, I think improved data quality. I think it's educational opportunity. If you think broadly beyond kind of science, I think doing data-driven project on all levels, from bachelor to the master to PhD, it's a great thing to do. And I think citizen science, right? Like as the broad community, many people in the world have access. So one thing is like what I think is receiving less attention, that data being available doesn't mean it can be reused, right? There is quite a bit of steps we need to do in order to reuse the data. So we were curious to know, do we do that as the scientific community? Are we doing data reuse? And if so, at what scale? And what are the trends there? So this is the project with Serghei University in Romania, as well as Technical University in Moldova, and the work done by Viorel and Nick. What we did, we took all the papers available on the PubMed. So that was around 6 million papers. And we processed that, and we identified the papers which do have GEO or SRA ID in the text of the publication. And this is what we call omics-based papers. But then we would like to classify the papers at the primary and secondary analysis. And that was quite challenging, because the text we use in the publication, when we say that we are sharing the data, or like we have generated the data versus we have used the data, is not that standardized, which was not a surprise. But that's the greatest challenge. If you look at the data availability statement in the paper, it's very hard to differentiate, or impossible to differentiate. So we're still working on NFF, and we're excited to try some LLMs to help us. But so far, we find that impossible to differentiate the papers on the text of publication. Is it primary analysis? So meaning the paper have generated the data versus the paper have reused the data. So what we did very simple thing. And we said the very first paper which has introduced the data, this is the primary analysis, and every other paper following that, that's a secondary analysis. So that's the assumption we did. So what we can see, and we look at a variety of data sets, meaning that we have looked at RNA-seq, we have looked at gene expression array data, and for RNA-seq, we have looked separately on gene expression data as well as raw sequencing data. So what you can see here on the left, that's gene expression data by microarray. So what you can see that it was, first of all, you see that was around for a while, but was not much data generation happening and not much data reuse happening. So at some point around 2020, around COVID times, you can see there are more papers reusing the data, generating the data for that technology. So to acknowledge that that's no longer popular technology to use. So there is a slowdown in data generation, but there is an uptake in data reuse. So even the technology is no longer relevant and to some degree, and people are not using that as technology of choice, people like to reuse this data. So if you think on the right, that's RNA-seq data, not just RNA-seq data, but the gene expression data sitting on Geo. So we can see, first of all, the data is quite popular, meaning that there is a lot of data being generated, but there is quite a bit of fraction of the secondary analysis happening. So if you think about other data types, and in this case, it's RNA-seq raw data on the right, and then whole genome data on the left. So in both cases, data reuse takes a substantial role in both, substantial fraction. So this is kind of to summarize it. So talking about microarray gene expression data, majority of the papers would be the papers doing secondary analysis, which to me is quite remarkable. And for other technologies, quite a substantial, almost 1/3 of all the papers are doing secondary analysis. Yet, we don't take advantage of the publicly available data, meaning there are many data sets which are in the public domain and never touched again, right? Like they have been generated, being analyzed, being uploaded to the public domain, yet were never being reused. I think it's open an interesting question. Why is that? And can we predict it? Because there's a lot of discussion that as we get more data, should we store every single data set? But then if we can use some features and try to predict the reusability, maybe we can prioritize or highlight the most reused data sets. So that's open interesting discussion. Not to mention that there is quite a cost behind the fact that we have instant access to the data. There is quite an operation and infrastructure behind which we don't fully take advantage. So there are some superstar data sets which are used over 10,000 times. And I think it raises the interesting question, what is more important? Is it what you do with the data when you generate it, or what the community will do with the data? And I think in this case, the answer is clear. The community have used it 10,000 times. So it's not just 10,000 attempts, that's rather 10,000 successful attempts if you think publication is an indicator of successful attempts. So there are more than 10,000 publications there reusing that data. So there is something interesting there. There is something specific there. And perhaps the value of data sets is in the community adoption or community reuse.
What happened in 2021?
COVID. People were bored, not being, I guess, able to go to the wet labs. Yeah, and then they turned, yeah, exactly. The trends are interesting. Exactly. Thank you, David. So yeah, I forgot to mention that. So yeah, so 21, yeah, something happened, but also, but maybe also the critical mass of the people being able to analyze the data, or people coming from computer science to bioinformatics. So it's also kind of a reflection of the community, or the critical mass of people being able to what is called, to analyze data, because this is the publication data. So maybe people started during the COVID, and by 21, 22 were able to publish it because there is a delay in the publication. It's also interesting to look across the world and see what is going on in terms of data reuse and data generation. I think data generation is more trivial. As there is financial resources, those parts of the globe can generate the data, or there is facilities, wet lab facilities. But data reuse is more tricky, or less, I guess, predictable. Theoretically, anybody in the world can reuse the data, but does that really happen? So this is some very preliminary results. I think if you think about Europe, for example, there is a clear differentiation between, I guess, Western developed countries and developing countries, for example, but there is other interesting trends where I think both US and China, for example, are leading in both data reuse and data generation. So there is more into that, and would be interesting to identify the data generation hotspots and data generation or bioinformatics hotspots of the world. And what we see so far, we were really interested to know, is there a country or region in the world which would skip data generation and immediately jump on bioinformatics? So far, we don't see it. We don't see a country which will substantially do bioinformatics skipping the data generation. So perhaps it indicates the value of the culture or expertise which you need in order to do the bioinformatics. It's not just, so you cannot kind of skip that. You need to have some critical number of researchers doing more wet lab or some domain expertise. So open questions, how to create incentive to create reusable data sets. I don't think there is currently enough incentive, or like people don't, so there is, because it's time consuming, and how we do that. So one idea we have got, we would like to introduce data reusability index, something which you saw on the plot showing the 10 most reused data sets, and maybe cherish the people who do the great job generating the data, have some leaderboard, and highlight, even that plot, highlight it broadly that there are some data sets which are extremely, extremely important for the community. I actually would be surprised, I mean, I would be delighted to email at some point those researchers who generated such cool data sets. I'm not sure if they know about that, right? So that's the data sets, hope they do. But would like to emphasize that the data set that they have generated maybe many years ago is so important for the community. Award and recognition program, right? So we should think about how we do that, how we can create reward and recognition for the people generating important data for the data reuse. Format matters, right? So what you saw that the format matters if you give people gene expression data, which is essentially a matrix that can be, I mean, not recommended, but can be downloaded in Excel and with no coding skills can be processed. Again, not recommended, but it's possible. As the raw data, it's quite a nightmare for the ones not into bioinformatics. You need to have access to high performance computing. You need to know what you're doing, many steps, select bioinformatics software. And many steps are redundant. If you think about that, what we do in terms of data storage, we store the data as the raw format, but maybe that's not, that's great to preserve the data for generation, but that's maybe not the base for data reuse. So think about FASTQ files versus BEM files. So in SRA, the data is stored as the FASTQ file, but what if I want HLA region only? I would need to remap the reads, and everybody perhaps doing that on the globe, doing redundant effort. So we need to think better about that. Okay, so to conclude, so what we see, data use is taking off. That's great news, and there is a lot of opportunities. There is also the warning there, and I just saw another day paper in Nature talking about papers which are doing reuse or not reuse, which use public health data, and many of them, let's say, reduce quality. So there is also the concern there that can unlock, especially given AI. So theoretically, you can do quick and dirty things and then not do enough rigorous. So we need to think about that as well. And for the future work, would like to look at other data types, including metagenomics, ChIP-seq, and other data sets, and see what's going on there. And we think about enhancing the utility of the data. As we have the data there in the public domain, one limiting factor is often metadata. The data can be publicly available, yet metadata is not there, which can prevent the reuse. But also we can think about enhancing the value of the data. If you bring additional phenotypes like HLA type in RNA-seq, which was originally not available, but can be inferred from the data, maybe that can enhance the utility of the data. And I think so to conclude, so data reuse is the cornerstone of open science, and I think it makes science to be accessible for all, regardless the country you're from or like in any part of the world. So I think that's really exciting. Okay, so that was the first talk. So I think, should I keep the questions towards the end, I guess, since a short talk?